Welcome to the second lecture on bacterial transformation. In our first lecture on bacterial transformation, we have already discussed about the findings of Griffith, Avery MacLeod that actually helped to understand that DNA is supposed to be the genetic material. We have gone through the steps of natural transformation and we have seen that it is the double-stranded DNA that initially binds onto the bacterial cell surface, and during its entry it gets converted into a single-stranded DNA. We have already seen that the single-stranded DNA undergoes substitution reaction inside the recipient bacteria, and this has really helped us to understand the steps of transformation. In today's lecture, we will be taking in details the various enzymes which are involved in making the cells competent and how these genes are regulated and how they are really executing the step of natural transformation. We will be discussing it in two parts. Number one will be discussed about the gram-positive bacteria and their mechanism of DNA uptake. Then we will discuss about gram-negative bacteria. To start with gram-positive bacteria, the model organism that we will be using is Bacillus subtilis. Herein, the genes which are responsible for the uptake of DNA are designated as COM gene, where COM stands for competence or the ability to uptake a DNA. So broadly, we can see that there are two types of regulatory COM genes called COM-A and COM-K. And there exists COM-E, F and G, which are responsible for making proteins that are present onto the cell surface or on the membranes and which are responsible for binding the DNA and uptaking of the DNA from the outer cell surface to the inner cell. So you will see that what is the function of COM-G? COM-G is responsible for formation of pseudopilus. The function of pseudopilus is to interact with the foreign DNA in an electrostatic fashion, whereas the negative charges of the DNA are interacting with the positive charged amino acids present on the COM-G proteins. Once the DNA is interacting with this pseudopilus generated by COM-G, then we will see the involvement of COM-EA protein. COM-EA protein will bind to the double-stranded DNA and it allows the entry of the DNA through this pseudopilus. And during its entry, it is the nuc-A gene that converts the double-stranded DNA into a single-stranded DNA. And we will see that during its passage, we will see the involvement of COM-EC, which is forming a channel, and COM-FA protein, which is acting as a ATP-dependent energy transfer mechanism. The DNA gets transferred from the outer cell surface to the inner cell surface. In case of Pseudomonas, we have seen that it is almost 10 kb of DNA is getting entry inside the bacterial cell. In case of Bacillus, we will see that almost 6 kb of DNA gets entry inside the bacterial cell. We have also seen that the rate of entry is almost 80 to 100 nucleotides per second, and in case of S.pneumoniae, we have seen that it enters with a 3' to 5' polarity and it almost takes 2 minutes for the DNA to enter.
So ma'am, do gram-negative bacteria also follow the same procedure of DNA uptaking like the gram-positive bacteria?
Yes, gram-positive bacteria follow a trend. Gram-negative are similar to it, but there is a slight difference, as because we all know that gram-negatives have an extra outer membrane, right? So the DNA has to cross two membranes, outer membrane, then lipid bilayer and all those things. It means that the negatively charged DNA, when it passes through the outer membrane, it feels a repulsion. So it has to be removed. That's why we need generation or formation of a pore, and that pore is generated by a protein called PIL-Q. So PIL-Q is also synonymous to type 2 secretion system proteins which form a type 4 type of pili, right? So these proteins are making a pore through which the DNA can pass. So they are not only responsible for bringing the DNA in close proximity, but they are also responsible for entry of the DNA through the outer membrane. So now we will start about regulation of natural competence. So in case of S.pneumoniae, we have seen that they show maximum competence in the early exponential growth phase. On the other hand, Bacillus subtilis and Haemophilus influenzae become competent when the nutrients are limiting. Whereas in case of Neisseria gonorrhea, we have seen that they express their competence under all the conditions. So herein we can clearly see that different bacteria have got different time periods of expressing their competence.
Ma'am, how is competence regulation brought about in Bacillus subtilis?
In case of Bacillus subtilis, we will see that it comprises of genes called COM-E, F and G operons, which are responsible for exhibiting competence. But these three gene expression or these three genes operons are switched on the basis of a transcriptional control element called COM-K. And this transcriptional element COM-K is under the regulation of COM-P, COM-A, two component systems. So COM-P is basically a cell surface protein which can sense that the bacterial cell Bacillus is running out of nutrients and the population has reached a high density. So it will sense this situation and it will undergo autophosphorylation. Once COM-P gets autophosphorylated, then it transfers the phosphate to its response regulatory element called COM-A, which is present in the cytoplasm. And when COM-A gets phosphorylated in its aspartate residue, then it carries on the phosphorylation of COM-K and it activates the transcription factor COM-K. COM-K is an important transcription factor which is responsible for switching on of COM-E, F and G genes, which are responsible for uptake of the DNA, as we have just seen previously in this lecture. So you will see that there comprises of two such elements. One is COM-K as well as COM-S. So what is the function of COM-S? That it protects COM-K from getting degraded, and thus it is allowing COM-K to act as a transcription activator. So herein we can see the involvement of two components, COM-P and A. One is sensing the signal and the second one is the sensor kinase that is accepting this signal inside the cytoplasm and transmitting this message to the transcriptional activator COM-K. So on the other hand, we need to also look into the basic relationship between the competence, sporulation and the other cellular states of Bacillus, as because Bacillus is capable of undergoing competence as well as sporulation. So when and where it will decide which mode to go is to be regulated, right? So when Bacillus subtilis reaches a stationary phase, the cell acquires competence and some others will sporulate. So at this stage we will see that there are two categories of cells. Some is acquiring competence, other is undergoing sporulation. So to coordinate sporulation and competence, Bacillus subtilis is producing certain regulatory peptides. One such important regulatory peptide is called PHR, and it regulates COM-A indirectly, inhibiting a protein called RAP protein. What is the function of RAP protein? It is dephosphorylating the COM-A protein. So by preventing RAP-A, we are preventing the dephosphorylation of COM-A protein. So like COM-X, we will see that PHR peptides are processed from the signal sequences of longer peptides, right? And what it does, the products of this, unlike COM-X, which interacts with COM-P at the cell surface, the PHR peptides are transported into the cell by the oligopeptide permease called SPO-OK. So, the SPO-OK gene is an example of a regulatory gene that is required for both sporulation and the development of competence. Now, we will be discussing about the regulation of competence in Streptococcus pneumoniae. This has been established just few months back. So, competence regulation in Streptococcus pneumoniae utilizes the cell-to-cell signaling by small peptide competence stimulating peptides called CSP, stands for competence stimulating peptide. So, binding of CSP to the membrane bound COMD sensor kinase results in phosphorylation of its partner response regulator called COM-E. Then the phosphorylated COM-E will activate transcription of 20 competence genes. Among one of them is encodes a RNA polymerase sigma factor. This is something interesting, that this newly synthesized sigma factor will interact with the core RNA polymerase and will dictate the RNA polymerase to recognize a new set of promoter sequences, and which are found upstream of 60 additional competence genes. So, in this way, by synthesizing a new sigma factor, the system has started synthesizing 60 new genes which are involved in the competence development. So, these late competence genes encode most of the process of DNA binding and transport processes. So, we know that bacteria basically are haploids, right? So, they don't have any sexual life. So, if they want to bring diversity in their genomes, that will help them to survive in a better way in the changing environments. The three options that are left for them is transformation, conjugation and transduction.
So ma'am, apart from this, what is the reason that bacteria participate in the natural transformation?
So there were various propositions. Initially, scientists were thinking that to undergo natural transformation, bacteria basically wants to get DNA as a nutrient, just as a source of carbon and nitrogen. But this was tremendously argued that if it is just as a source of nutrient, then why at all certain bacteria like Neisseria are uptaking their own DNA only? If it is just a case of nutrition, then they should have taken any type of DNA, right? So this argument was further argued by people by saying that by uptaking its own DNA, the bacteria is quite safer, because if it uptakes a foreign DNA, then via this foreign DNA, some prophages or some transposable genetic elements which might be containing certain genes which are responsible for killing of the bacteria may enter the bacterial cells. That's why bacteria has chosen to uptake its own DNA. Apart from this, there are certain evidences wherein we have seen that nutrition, it is utilizing it as a nutrition, fine, but in case of DNA damages which are occurring by UV rays, that pyrimidine dimers are formed in the DNA of the bacterial genome, and to repair its own DNA, it tries to go for a option of transformation, wherefrom it procures a self DNA which may not have got damage in the same region, and by this means it can repair its own genome. In this regard, people were arguing then that competence development should be directly proportional to UV damage or DNA damage. On the other hand, the repair mechanisms should also be switched on along with competence. So similar phenomena was observed in case of Bacillus subtilis and Streptococcus pneumoniae, wherein they could find that the Reg A gene, which is responsible for recombination, are switched on in presence of development of competence. So we can infer out that competence development is leading towards the repair mechanisms are switched on. Or in other words, we may say that UV irradiation is creating damages in the DNA, and to repair that damage, it is developing competence, and by competence acquiring, it acquires the foreign DNA, and that foreign DNA, when gets inside the bacterial cell, it has to be replaced with the damaged DNA, taking help of the repair enzymes. And the third option of natural transformation was that it undergoes recombination events to bring diversity in their genomes. That diversity might help them to evolve in a better way in the changing universe. So these three were the proposed possibilities for bacteria undergoing natural transformation. Now we will be discussing about the concept of mapping of the genome utilizing the phenomena of transformation. In this regard, it was proposed that co-transformation frequency was calculated. Say for example, there exists two marker genes X plus and Y plus. If the probability of entry of X plus independently and probability of entry of Y plus independently is calculated, and probability of these two genes getting entry together is more than the product of their independent entries, then it can be concluded that X and Y genes are close together. But in this regard of calculating the gene order on the basis of co-transformation frequency, there exists a problem of congregation. What do you understand by congregation? Congregation basically is that a bacteria has got more than almost 20 receptors on its cell surface through which the foreign DNA can get entry by the method of natural transformation. So it may happen that two different marker genes are getting entry inside the recipient bacteria, but they are entering independently through two different receptors. And we might be thinking that the two markers are getting co-transformed together, and we will be concluding that they are linked together, which is not the case. So to go for a mapping of genome utilizing co-transformation frequency, what should be done is that the amount of DNA that is being added should be limiting, so that under none of the circumstances a bacteria receives more than one piece of DNA at a time. So these are the points that are to be taken into account while carrying on mapping of the genome utilizing co-transformation frequencies. We have discussed that co-transformation frequency can be used to calculate the gene map of a bacteria.
Ma'am, several bacteria participate in natural transformation. So can this process of transformation be induced even artificially?
Yeah, this is supposed to be one of the major applications of bacterial transformation, that we can artificially induce competence among those bacteria which are not naturally transformable. There are various methods of inducing artificial competence. Number one is by treating the cells with calcium chloride. So this methodology was first established by the scientist called Stanley Cohen in the year 1973, where he said that if the cells are grown to an exponential growth phase and their growth is stopped there, and these cells are treated with calcium chloride, and this calcium chloride treated cells, when treated with any plasmid DNA and heat shock was given, followed by chilling on ice and a recovery phase, led to introduction of the foreign plasmid DNA inside the bacterial cell. This is supposed to be the most simplest method of introducing any plasmid DNA inside a bacterial cell by artificial means. which helps us to clone gene of our interest inside the bacterial cells. Apart from this, if the bacteria is of gram positive in nature, which has got a thick peptidoglycan layer, under such circumstances calcium chloride mediated artificial transformation may not give fruitful results, so we may go for electroporation, wherein the transformation efficiency is also very high. The basic principle of electroporation is that we introduce the cells inside cuvette between the two electrodes. The cells are made competent by treatment with glycerol, and electric pulses are given, and the cell which is mixed with the DNA during the course of electric pulse, the foreign DNA gets entry inside the bacterial cell. So the basic principle is utilizing electric field to introduce foreign DNA or plasmid DNA inside a bacterial cell. Apart from this, we have got another method called Protoplast Fusion, wherein the cell wall is removed. After that, what we see that the protoplasts are basically susceptible to osmotic lysis. So they should be kept in a buffer which is osmomolar with the cytoplasmic molarity. After that, we will be adding DNA and polyethylene glycol. What polyethylene glycol does, that it helps in the membrane fusion, and during this course the DNA gets entry inside the cytoplasm. After the introduction of DNA inside the protoplasts, what is done is that we carry on steps which help in regeneration of the cell wall. So these are the three important methods which are used in our labs for artificially introducing any DNA inside a bacterial cell.